All right. Good afternoon, everyone. It's my absolute pleasure to introduce today's speaker for another round of our boundary breaking science lecture series. So today we have Dr. Dasha Nelidova from the Institute of Molecular and Clinical Ophthalmology in Basel. And Dasha's lecture is a part of an overarching lecture series from our 2021 Early Excellence in Science awardees. Now, this award goes to young breakthrough talents having already huge impact on their respective fields in four areas: biology, chemical and data sciences, as well as medical science. And today's speaker, Dasha, is the winner for the medical sciences category. As I mentioned, this is an overarching series of lectures, which really aims to build a bridge between our prize winners and also our broad scientific network that includes academic experts, industry researchers, physicians, as well as other early career scientists. And most recently, we had Connor Coley, one of our recipients, as well as Marika Udelar, right? And today it's, of course, our pleasure to have Dasha here with us. Now, before introducing Dasha, just a couple of housekeeping points. Use your Q&A tools in Zoom to ask your questions. I'll be there to monitor it and cluster a bit these questions. And yeah, after this session, the recording will be available at the Bear Foundation team space, as well as at the YouTube channel of Bear Foundation. And we do encourage you to join us as well afterwards for an open discussion following Dasha's talk, and there would be a separate link to this invite then. Good, without further ado, now, again, it's my absolute pleasure to introduce our speaker, Dr. Dasha Nelidova. So as a native of Switzerland, Dasha received her bachelor's degrees in medicine, surgery, and human biology from the University of Auckland in New Zealand. And Dasha is one of the unique breeds of medical doctors and researchers, because afterwards, he pursued her PhD at the Friedrich Miescher Institute for Biomedical Research in Basel, really with the highest recognition there of summa cum laude. Now, her work on the state-of-the-art methods to restore vision were really recognized by several accolades and also published in prestigious journals, including the Science magazine. And to name some of her numerous awards, Dasha is a recipient of the Science and SciLife Lab Prize from the Young Scientist for Young Scientists. Dasha is also a recipient of the Röntgen Prize from the University of Gießen, named after the discoverer of X-ray. And recently, Dasha has also been awarded, of course, in the name of this talk, by the Bear Foundation as an early excellence in science awardee for the medical sciences. Now, Dasha's work in bringing together gene therapy and nanotechnology to address retinal degeneration is really it's fundamental, right? Because it is retinal degeneration is a key leading cause of blindness, and we are going to learn about that today. I'm really excited about that. And in her current role, Dasha is a research fellow of the Institute of Molecular and Clinical Ophthalmology in Basel. And with that, without further ado, I'm going to hand it over now to Dasha then for her talk.
Thank you very much, Michael, and thank you for your kind invitation to speak here today. Let me see if I can share my screen. I think it's now up on the screen for everyone, right? So I will tell you a story about light, gold, snakes, and the retina. And it starts in 2010. In 2010, a beautiful paper was published The subject of that paper was optogenetics, a new kind of gene therapy. And the aim was to show that the optogenetic method can also be used to restore vision in blind mice with retinitis pigmentosa, which is a rare, aggressive, and inherited type of retinal degeneration. It took our field a few more years to get to the next stage, because the next stage was clinical, a case report. This time, a different optogenetic sensor was targeted to the retina of a blind patient who had advanced retinitis pigmentosa and minimal remaining vision. Because of technical reasons, the sensor was actually targeted to the last layer of the retina, this black so-called ganglion cell layer, and not the first layer of the retina, this purple photoreceptor layer, which is the layer normally responsible for sensing light, and the layer actually affected by the disease. Nevertheless, researchers from Basel, Paris, and a center in the United States showed that the patient could perceive, count, and touch different larger objects using the treated eye, while also wearing specially designed eye goggles. These objects included a large black notebook positioned at the center of a white table, a smaller black box of staples, as well as detection or discrimination of multiple objects, a series of black tumbler glasses. For the very final visual task, object detection as a yes or no task was paired with multi-channel EEG recordings. So recordings where electrodes are attached to the back of the scalp to prove image-driven activation of visual brain regions. This result was obtained around after 12 months after the patient received their eye injection, and after around 15 visual neurorehabilitation training sessions, where the patient was given different kinds of exercises. So first, exercises to direct or maintain their gaze in order to identify the direction of the object. Then combinations of head and eye movements to better scan targets. And finally, simple hand-eye coordination exercises to reach forward to grab objects. But really, because every patient, every case is different, the exact kind of neurorehabilitation strategy required has to be tailor-made for that individual. Interestingly, outside, the patient reported that he could see zebra crossings or pedestrian crossings. So in the future, the ability to navigate might also be tested. One group has already designed and validated a special obstacle course for patients with retinal disease who are receiving another kind of retinal gene therapy. So here the researcher changes the amount of light that's available in the room, going from light to progressively darker. And then patients are scored on their ability to avoid obstacles while also maintaining a reasonable speed of navigation. These kinds of visual tests are non-traditional, but they are nevertheless of great interest to rehab specialists because they're likely better suited to evaluating recovery in patients who have low vision. Recently, data from several additional patients became available, also showing an improvement in visual function. And because of these promising results, optogenetic vision restoration was granted fast-track status by the FDA. This does not mean that regulatory approval has been fully granted, but does indicate that we are on our way. One caveat here is that optogenetic vision is not perfect vision or natural vision, but rather it's designed to bring back some ability to see edges and contours, resolve different shapes, and see different larger objects, all perceived as black and white images. On a molecular level, optogenetics is a technology that sensitizes neurons to different wavelengths of visible light. Blue, green, orange, red, so different wavelengths of the visible spectrum, all by transfer of single genes that code for light-sensitive proteins. These genes originate from very simple organisms, usually green algae or bacteria, and are then transferred to strategically important retinal cell types using viruses or virus-like particles. 9 times out of 10, we use a modified adeno-associated virus, or AAV for short, which is a benign FDA-approved transfer vehicle. This technology works because the simple genes lead to the production of simple, meaning single component, light-sensitive ion channels. These essentially replace the multi-component phototransduction system that's normally found within the human eye. And so when light is switched on, current starts to flow through that ion channel, through the retinal cell, through other cells in the retina, and towards the brain. Light-induced photocurrent is the mechanism by which we see. This is the mechanism by which vision normally works. And so when this ability is lost because of disease, vision is also lost. So for us as vision restoration researchers, making a blind retina see again means finding a strategy to bring those retinal photocurrents back. In practice, we do this by combining gene therapy and surgery. We package these light-sensitive optogenes into special virus-like particles. And when manufactured, the virus just looks like a clear fluid. A few microliters of this can be injected into the eye, placed in front or behind the retina. In the photograph on the right, you can see a human retina, the retina of a child, being injected. And here, the retina is being lifted off as some of the viral fluid is injected underneath, making a bleb. With time, the fluid is resorbed and the retina flattens back down to its normal position. But importantly, the gene has been delivered. And then using a special mechanism, it makes its way to the correct cell type. For the optogenetic system to work in a patient, the patient also has to wear specially designed eye goggles. These capture the visual scene using video camera technology, convert the scene into an image composed entirely of one single predetermined wavelength, turn up image intensity quite a bit, and then project this high intensity image onto the retina, onto the optogenetically transduced retinal region by a system of several thousand micromirrors. The mirrors are in the rectangular array, each corresponding roughly to a single pixel. And then by flicking the mirrors up and down or on and off very quickly, and thereby adjusting how much light is allowed to go through, image patterns and light intensity grayscales can be produced, giving pictures a little bit like this pixelated Mammalian. And in real life, the goggles look like this. In clinical trials, as a precautionary measure, their use was limited to a maximum of 4 hours per day. But optogenetics was well tolerated in that small cohort of patients with no major adverse effects, as reported by the lead clinician in conference proceedings. A key issue with this approach is that optogenetics requires bright, extremely bright visible light in order to activate. Quantified, this is at least 10 to the 16 photons per square centimeter of the retina per second, which is a lot of photons. So in practice, this would mean flooding the human eye with light, leading to photobleaching, to the sensation of glare, to saturated photoreceptor responses, all of this interfering with whatever vision the patient has remaining. And since most of our patients, including those with age-related macular degeneration, still have some degree of remaining visual function, which should be preserved, there was strong pressure in our field to develop a new class of technology. In age-related macular degeneration, cone photoreceptors of the very central retina lose their light sensitivity. Surrounding photoreceptors remain mostly viable, and in most cases, peripheral vision is largely unaffected. But at the present time, after patients become centrally blind, age-related macular degeneration has no effective treatment. We described a new method using near-infrared light, nanogenetic light sensors, designed to be compatible with remaining vision for the patient group with incomplete or regional photoreceptor degeneration, such as age-related macular degeneration. And the advantage of near-infrared is twofold. First, in most species, including humans, vision is detecting or is sensitive to electromagnetic radiation that is in a pretty narrow band, so roughly 400 to 700 nanometers. And near-infrared light at around 900 nanometers is outside this window, meaning that it is not expected to excite remaining photopigment. And second, water absorption of near-infrared wavelengths is low, on par with water absorption of the visible spectrum, and lower, much lower than water absorption of longer wavelength infrared radiation. Since cells and tissues are mostly water, this means a better safety profile, as the near-infrared pulse is traveling through the eye to reach the retina, since it won't be absorbed, so will not transfer energy to tissues that are at the front of the eye. The idea to use slightly longer wavelengths actually came from snakes, because snakes can see the world in two different ways. Like humans, snakes can use their eyes to detect wavelengths of the visible spectrum. In addition, certain species can also generate thermal images. These snakes detect long wavelength infrared light, so wavelengths between 5 and 30 microns, using temperature-sensitive TRP cation channels that are expressed in a specialized pit organ structure. This is a shallow depression or concavity situated about halfway between the eye and the nose. And then importantly for neuroscientists, thermal and visible spectrum images then superimpose within the brain, presumably enabling the animal to react to the environment with greater precision than what is possible using just one single image. It's thought, based on some work done in the 70s, that snakes can switch back and forth between these two imaging systems, or else use both simultaneously. I always thought it was a bit unfortunate that the human visual system does not have a similar inbuilt backup plan, but it is possible to borrow a few ideas from nature. We cannot really use long wavelength infrared light for the human eye because there is a risk of overheating ocular tissues. We can use near infrared light because it's cooler, although this same feature actually makes it an inefficient activator of TRP channels. To develop a more efficient near infrared light detector for retinal cell types, we engineered a dual system consisting of a genetic and a nanomaterial component. The genetic half of the sensor was a temperature-sensitive TRP channel, but engineered to incorporate an extracellular protein epitope tag, shown on the screen as orange, that was then recognized by a specific antibody. The nanomaterial half of the sensor consisted of gold nanorods conjugated to antibodies against the epitope. This system uses surface plasmon resonance and electron oscillation along the nanorod surface to convert near-infrared light into a small amount of local heat, which causes photocurrents to flow through the TRP channel. We saw that expression of this nanogenetic sensor in the retinal cone photoreceptors rendered blind mouse retinas near-infrared light sensitive. Cone photoreceptors, which represents retinal input, and ganglion cells, which represents retinal output, responded vigorously to near-infrared light. You can see that this calcium trace is shooting up every time a near-infrared pulse is delivered, whereas in untreated animals, the response remains a flat line. These retinal responses propagated from the retina to the brain, which allowed treated animals to perform simple behavioral tasks. In this task, on the left, a mouse takes a drink of water every time a near-infrared pulse is delivered to the eye. Near-infrared sensor injected mice learned to associate light with water within 4 days and performed this task as well as healthy wild-type animals that were trained for 4 days using visible light. In complementary experiments, we confirmed that near-infrared light neither activated healthy wild-type cone photoreceptors nor affected their subsequent visible light responses. And similarly, awake wild-type mice failed to exploit or pretty much ignored near-infrared light cues during behavioral training. We already knew that nanorod properties depend on size and shape. And so by using slightly longer nanorods, going from a length of around 80 nanometers to around 120 nanometers, we were able to tune near-infrared vision to another near-infrared wavelength, going from 915 nanometers initially to 980 nanometers. This might seem like a small change, but wavelength tuning is important for several reasons. First, certain wavelengths might be better tolerated by patients than others. Second, maximum permissible exposures or light doses for the human eye depend on the wavelength. And so the more redshifted, the more photons we are allowed to deliver. And lastly, enabling near-infrared light sensitivity within a blind human eye requires eye goggles that utilize specific wavelengths. Compatibility with our current, but also our future near-infrared projection systems requires tunable near-infrared light detectors. In follow-up experiments, we showed that ion channels, epitope tags, antibodies, these components can all be modified or exchanged to gain additional desirable properties. For example, we selected TRPA channels from the Texas rat snake because of their lower thermal thresholds, and compared to mammalian TRPV channels, we saw that mice with a snake channel were better able to perform their behavioral tasks when lights were dimmed, indicating an improvement in sensitivity of the sensor. Finally, we validated findings in blind ex vivo human retinas. To do this particular experiment, we had previously developed a cocktail of molecules that could keep human retinas alive up to, it used to be 10 weeks, but now it's closer to 14 weeks post-mortem, which gives us enough time for gene expression in the human retina to take hold. We targeted our nanogenetic sensor to human photoreceptors and saw fast, strong activation of human photoreceptors and all the downstream retinal neurons, appropriate downstream retinal neurons, including ganglion cells. Taken together, these experiments provide proof of principle for potential therapeutic translation. Light intensity is required to drive the system, met existing safety standards that specify exposure limits for the human eye, and we further demonstrated that components might be exchanged with predictable final outcomes. So in the future, targeted central repair would potentially allow an island of near-infrared sensitivity to be built surrounded by a sea of natural vision, or parallel developments in surgical techniques, and also near-infrared light projectors with eye tracking capabilities for the human eye make targeted central repair feasible. Ultimately, the user might be able to self-select the region of the electromagnetic spectrum that is most useful to them to view the external world, a decision that's guided by the state of their retina and also ambient light conditions. Thank you very much for listening today.
All right. Thanks a lot, Dasha, for such an enlightening talk. Yeah, no pun intended there. Yeah, but really, yeah, quite a learning from my side. Good. All right. So just reminding the audience, virtual audience here as well, to please post your questions on the chat box here for Dasha. But maybe I could start while waiting for the audience questions here. So really, really cool approach here, Dasha, combining genetic techniques there, right? Also with the nanorods. I'm just wondering, in the mouse models, and perhaps you could also comment with the human retina system here, what's the threshold of photoreceptor cells that need to express these channels, temperature sensitive channels, yeah, basically the construct, so that you get significant or measurable response from mice, or also perhaps yeah, with ex vivo system.
So from memory, I think we reached about 20% targeting efficiency, and we could already detect responses, not just in single cells of the retina, but also in the brain. But if you can drive this up, that would be even better. The way to do that would be to design gene expression cassettes, including promoters that are able to drive stronger, high level protein expression.
I see. Interesting. And what about also driving it down, maybe to have sort of, I mean, driving it up, of course, to up the response, but perhaps, you know, there are cases where what's the base sort of threshold where there's already significant impact there in cases where maybe efficiency, transduction efficiency is not so high. You know, would we already see impact, for example, for 10% expression of cells, or?
Well, it's possible. So if you have a lower transduction efficiency, it might still be sufficient to bring back some sensitivity to light. Ideally, we want to make as many light insensitive photoreceptors light sensitive.
Makes sense.
And our second aim would be to then improve the sensitivity of that system to photons, meaning that we would require fewer activation photons to produce a certain number of spikes in ganglion cells. But this is a question regarding the activation threshold of that particular channel.
Makes sense, makes sense. I'm just also thinking into, you know, in humans, right? When this, of course, hopefully, of course, becomes a reality, how do you envision also the administration there? You know, do we need to redose the transgene? Is it stably expressed throughout, you know, the lifetime? Or the nanorods, for example, is that something that you also need to administer every month or so? Or just thinking about the clinical sort of application of such system?
So for delivery of light sensitive genes to the human eye, we're learning the most right now from the visible light optogenetic clinical trials. There are around 8, or perhaps it's more now, patients who have received that therapy, and they are telling us what they can see, which then suggests new ideas to researchers in terms of what improvements are possible. And there was a second part to your question.
The dosing piece.
The dosing.
Yeah.
So the first decision that we have to make is what cell type to target, because it's possible to target to the last order neuron on the retina and still show that the patient can see something, that there is recovery of partial vision. The next step for the field would be to target to light insensitive photoreceptors if those cells are available. And then in terms of dosing, for the gene, it's not possible to say exactly. We still don't have a good way of predicting the mean and variance of gene expression in the human eye. But we are getting better at this by testing in ex vivo human retinas, but this is ongoing. And then in terms of nanorods, so actually one modification we are working on right now is to make everything 100% organic. So that might change how the sensor looks quite significantly in the end.
Good. No, thanks for addressing that, Dasha. So there's a question in the chat. So about the method of restoring light sensitivity here, right? The question is about how many patients would benefit from such a technique, and how many have lost too many photoreceptors to be eligible for this approach? Think
Yeah.
about the patients in the end.
So there are a lot of patients with age-related macular degeneration. It's maybe 1 in 6 or so after a certain age, so over 60 years old. It is then possible to select from this large group of people certain cohorts who are eligible for this sort of approach and who perhaps would benefit from another approach. Some patients may not have enough photoreceptors for photoreceptors to be targeted, but may nevertheless benefit from this approach if the next order neuron of the retina is made into an artificial photoreceptor. So that would be one way to think about it. Either if you have photoreceptors, that's the cell type you target, and if you do not, then you go to another appropriate downstream cell type to bring back some ability to see large objects.
Good. Thanks, Dasha. There's also a question here on, maybe could you elaborate a bit why gold materials are used for the nanorods here?
It's one of the best studied materials that has this photothermal capacity, a relatively high efficiency of light to heat conversion. It's also a noble metal, so it's more biocompatible than other materials with the same property. And finally, it's easy to functionalize them with antibodies due to some surface chemistry that makes this possible. So you can potentially improve the specificity of your targeting.
Good, thanks. There's also an interesting question here about the scope of the application of such state-of-the-art technology. Is it possible to apply this technique to various kinds of blindness, or is it specific for example, a partial loss of vision or only a specific type of blindness here?
It's not specific to any disease. It's designed in a way to make it compatible with remaining vision, should the patient have remaining vision. It works best when we target the center, or at least one region of the retina, because for technical reasons, we cannot at the moment achieve retina-wide coverage. We can only physically deposit our virus and or nanocomponent in one small region of the human retina. But in terms of the exact disease, the mutation, inherited or acquired, this doesn't play such a huge role. It's more that the state of the retina has to be characterized, and we would need one region where sensitivity is lost.
Good, thanks. There's also a question here about sensitivity. What would it require for the method to discern even smaller objects, to get more visual precision here?
So resolution depends on two factors. The first factor is the property of the sensor, for example, how fast it is to respond to light onset and also light offset. But the other bigger consideration is also the state of the patient's retina. If their degenerative condition is aggressive and they're losing cell types, despite the fact that we might have an excellent sensor, photoreceptors might not be available, for example. And the resolution that it's possible to produce when you go further along the neuronal chain, it will probably be lower. So to see smaller objects, the answer to that question is we would need to combine a strategy that brings back sensitivity to light with a second strategy that slows down degeneration in the retina.
Good. Yeah, really lots of interesting questions for discussion here. All right. Anything else from the audience? Yeah. Going once, going twice. But I guess Dasha, with your presentation, really addressed a lot of these burning questions as well, and the remaining ones here in the Q&A. And with that, we really do thank you once again for such a very, very informative presentation, and really looking forward, to seeing the future of this, right?
Thank you very much for inviting me.
In need as well. And again, um, actually, I see that there's still a question here. I would actually recommend also the audience for additional questions. Let's move in then to the separate link where we could have really this more thorough discussion on the topic. Yeah. All right. And on that note, yeah, thanks once again, Dasha.
Thank you very much.